So there are 6,000 to 10,000 genetic diseases that humankind is suffering from. That's a huge number of diseases, and ever increasing with advances in science. However, these are only diseases that affect a single gene and cause then a disease. There are much more diseases that have a genetic background. And when taking all these diseases together, we have 6 out of 10 people suffering from diseases with a genetic background. That's a huge number. And I'm not only talking about rare diseases like cystic fibrosis or sickle cell disease. I'm talking about very common diseases like diabetes, cancer, Alzheimer, diseases all of us know. When thinking about a cure for these diseases, because they're genetic, one automatically thinks about gene therapy. These are therapies that actually correct the genetic mutation. And looking or having a close look to these therapies, there's only 0.01% of diseases that can be cured with this approach. So you see there's a huge gap of patients that require improvement of curative treatments for them. So now I talked a lot about DNA, RNA, genes, genome. I'm pretty sure that you're all familiar with these terms, but let me take you to the kitchen to explain basic biology. So in this kitchen, we want to bake the best cookie, and we have this ancient recipe book from our grandmother where she collected the best recipes ever. And we want to make the cookie, but because it's so ancient and very precious, we don't want to carry it around the kitchen, so we make a copy of the best cookie. And here you see its typical recipe with sugar, butter, flour, cinnamon, and chocolate. We take it to the kitchen, assemble all the things, put it in the oven, and as a result, we have the perfect cookie. So what happens if there is a mistake in the recipe book from our grandparents? Well, we have the mistake in the recipe book. We then have the mistake in the copy. So instead of 1 cup butter, we have 1 cup of oil. We put everything together in the kitchen again. And as a result, we have a destroyed cookie that is crumbly and cannot be eaten properly. So, but we want our perfect cookie. What to do about that? So option number 1 is taking a corrector pen, opening this ancient recipe book, and correcting the mistake that is in there. As a result, we have the correct copy with butter instead of oil. We then assemble it again in the kitchen, and we have the perfect cookie. However, when opening this recipe book, the pages are so thin that when we corrected it, we automatically also wrote something on the next page, which is the best cake page. And instead of there having oil, we have no butter, and as a result, we have a destroyed cake. So now the problem is, as we corrected it in the recipe book, it is now corrected forever, or also falsely corrected forever, and forever we will have a destroyed cake. And therefore, correcting the book can cause permanent and unwanted changes that we cannot correct later. A better option would be to not correct the recipe book, but to correct the copy, which is easily done. We have it on a sheet of paper, we easily can correct it, and as a result, we have the perfect cookie. And also, it's a bit more flexible. So if we decide we want to have maybe cookie crumbles for our cereals, we can leave the recipe as it is. We are very flexible in changing it or not changing it every time we do this copy. So you might have already guessed so, but in basic biology, the recipe book is DNA, where the genetic code of life is written in, which basically makes us us. And to get the information from the book into the kitchen, we have RNA, which is a mobile form of genetic information. And this mobile form of genetic information we bring into the kitchen, which is the ribosome. And there we can assemble everything together, which is then as a product we have the protein. In this example, mistake is the genetic mutation in the book. And the corrector pen on the DNA level is gene therapy. But what I'm interested in my research is actually the corrector pen on the copy level, on the RNA level. And I would like to take you, go a little bit deeper to explain what I'm actually doing in that regard. So we want to change the genetic code temporarily on the copy RNA level. So let me show you here a string of letters, how we see it in the DNA, which is adenine, cytosine, guanine, and thymine. In the case of RNA, it's substituted with uracil. So we have this string of letters that encodes maybe for protein that is very important, and if it's mutated, we get early onset Alzheimer with the age of 40. So now we have this mistake here, the A instead of the G, as you see here. So there's the mutation, and when we transcribe it to RNA, we keep the mutation, and as a result, we have a destroyed cookie or patient that gets early onset Alzheimer with the age of 40. What do we do about that? Well, we take again the corrector pen, correct the RNA, and how do we do it? We don't streak out the letter, or we don't remove the letter or substitute it, but we take a mask and put it on the letter A, meaning that it's now not visible as letter A anymore, and our body would interpret this masked A as a G. And voila, we have exactly the same information we had in the original healthy version of the gene. And as a result, we have our perfect cookie, or a patient that doesn't get early onset Alzheimer, but stays disease-free till late age. So what is this corrector pin? What are these masks, these disguises on the RNA? Well, the corrector pen is an enzyme. You might have heard of enzymes. For example, we need enzymes to digest our food. And in the human cell, enzymes are like little hands. They execute tasks. They, for example, transport molecule A from this position to that. Or they might transform molecule A into molecule B. So they have little tasks in the cell they execute, and what we're interested in is the task to put the mask onto the letter A. And this is exactly what is happening in the cell. They take the mask, put it on the A, and we have a disguised A letter. So enzymes add disguises to RNA in the cell. And of course, it's not little hands that are everywhere in the cell, but it's a highly complex protein structure, like you see here in an example, which is specifically designed by evolution to exactly execute this single task in a cell. And in my work, I want to engineer these disguise enzymes towards certain goals. For example, I not only wanted to disguise the letter A, but I want to modify the letter Z, G in U as well. I wanted not to only add, but also remove, read out, or change the disguise on the RNA letter. And finally, I not only want to change it, but I want to have an output. For example, to produce more protein from a single RNA. In the cookie analogy, that would be not only making one cookie from the same recipe, but making hundreds of cookies for a whole party. So this is the goal, and this is why I'm engineering in my postdoctoral research the disguise enzyme. So why do we care at all? So right now, if a patient is sick, they go to the doctor, potentially get current therapeutics that include gene therapy, and some of them will be healthy at the end of the day. What my vision is that with RNA modification-based therapeutics, including these RNA disguises, but also other forms of RNA therapeutics, we will be able to help more patients and also cure them. And at the end of the day, this improves the care, especially for the diseases that affect so many of us, like Alzheimer, diabetes, or cancer. Because with these kind of approaches, we not only permanently change the genome, but suddenly have tools in our hand to do subtle changes that are necessary to cure these diseases. Thanks for your attention, and I'm looking forward to your questions.
Questions? Someone?
Thank you, Monika. That was a great presentation. I was just wondering how, in this specific type of strategy to treat these diseases, how do you overcome the issue of penetrance, of how much of the copied recipe you can actually turn with this enzyme? Do you aim to changing 100% of the RNA, or how much of it do you need? And what are the implications on the amount of doses that you need to provide to the patient? Is it sustained for the rest of their lives? What would be the implications of this? Thanks.
That's a very important question, really to the core of the difference between gene therapy and this RNA modification or RNA-based gene therapies. It is that we have here the advantage to only temporarily change or only change it a little bit. So there are diseases where you want to keep this protein or this gene. You don't want to switch it off completely like with gene therapy. And exactly for these diseases, that's the better option, because you can change 5 to 10%. Sometimes that's enough. You can decrease or you can increase. And this is the advantage it has over gene therapy. And as I said, it sometimes only requires a 5 to 10% drop or increase, not more. But you're right, you have to usually give it subsequently for several doses, not only one, like for gene therapy where you change it completely, but you have to keep giving this. But this is also often safer for the patient because you kind of decrease unwanted side effects.
Hi, thank you for your presentation. So I'm completely out of the field, so just a couple of curiosities. The first is whether these proteins that you're engineering can work on a general level, or if they are specific for some kind of illness. You mentioned Alzheimer many times, so maybe they're related to that. And the other question is whether they work only on the cure level, or if you can do also like prevention with this technique. Thanks.
So I come to the second question. I think especially for those diseases which are not prevalent right now, but where you know you will have it later if this is continuing, it could be used for prevention. Also because you don't have major changes, it's only changing it a little bit. But this is very visionary. There's right now no therapeutic out there that could prevent a disease with these approaches. So we're really at the beginning. And regarding your first question, the enzymes, they're usually very specific. So they execute one task, for example, putting this guys on the letter A. And it's the task of the researcher then to take this very specific enzyme and put it into a certain therapeutic context, or to make it less specific or even more specific and kind of tailor it to do certain tasks. So this is what people do when they do protein engineering, to change this a little bit.
So first of all, congratulations. It was a great lecture. Just regarding like how to regard the envisioning the how to target the specific cells that you are aiming to treat, let's say. I was just curious to know if you thought about how to specifically get to the cells that you want to treat it. If you want to treat Alzheimer, I guess that you need to get, in this case, to the neurons, or if you want to treat another specific disease. So if it's maybe a bit too far from what you're studying, or if you're considering like a way of getting to specific cells.
So specifically for my research, this is not part, so delivery strategies are not part of the research. It's more really the basics to figure out how we can use these enzymes in therapeutic context. But thinking about the COVID-19 vaccine, there we got an mRNA in a lipid nanoparticle that then expressed the protein. And enzymes are proteins, so if we want to express them in a cell, we could imagine a similar delivery strategy with nanoparticles or RNA, because proteins itself, to get them into the cell or to a specific region, you might know it from the blood-brain barrier, is really hard. So I think right now, with the success of the COVID-19 vaccine, this is the delivery strategy I would think of as most likely.
Thank you for excellent talk. Maybe it's for the for the future, question for the future, do you think that it is possible to combine two enzymes at once? Because, for example, some diseases are caused not only by one mutation. We need to do something with 2 or 3 proteins or more. So do you think that it will be possible to combine 2 or 3 enzymes in the same, let's say, dose? Or we need to treat every enzyme in every, let's say, therapeutic set?
That's very futuristic, and of course it would be amazing to have like a tool where you can correct 10 mutations, because you might know things like diabetes or cancer, they not rely on one single mutation. But right now I think the problem first is to get one enzyme into the cell, or leverage one specific enzyme that is already in the cell for a specific purpose. There are ways to do that, but then correcting several ones, in theory it should be possible, but we are far from that. So I guess there are decades of research still needed to really correct each and every single mutation there.
Okay, thank you.
Thank you.